Alright guys, today we're going to talk about the nervous system and how specifically your neurons function. Neuron function is all about transport. It's all diffusion and active transport. So it makes sense to talk about it while we've been looking at other parts of membrane transport. This chapter in your book has a lot of stuff about the different parts of the nervous system. It goes into a lot of depth about the different parts of the brain. Please don't concern yourself with that. It's not important. What the major thing that you want to look at is how do nerves function and why does that make us work the way that we work. So let's go through it. So why do we need a nervous system? Well, you've got a bunny, you've got a prey, and then look, no more bunny. Poor bunny. So your nervous system has to be fast so that you can respond when there is danger. It has to be accurate so you can run quickly when there is danger. It has to reset quickly so after you have run from something you don't freeze in panic. Okay, that would be bad because when you freeze in panic, you're easy pickings. Alright, so the nervous system is broken down into specific types of cells. The specific type of cell in the nervous system is called a neuron. Okay? It's got some different parts. There's the cell body, big middle part of the cell, and it's got all the organelles in it. Okay. Then dendrites, which are these big branches that stick off from the cell body. Then an axon, which is like one long tail that comes out. That axon is surrounded by something called the myelin sheath, and we'll talk about what that is in a bit. And then the signal is going to travel from the dendrites. It's going to come into the neuron at the dendrites, move down the axon all the way along this axon, and then it's going to move from here to the next spot through these synaptic terminals. Okay? And it may be going, these synaptic terminals may meet up with another neuron, they may meet up with a muscle cell, they may meet up with another, with your brain. Okay? Alright, the structure of a neuron fits its function. Remember, we're going to keep talking about structure and function all year long. There's many, many entry points for a signal. So one neuron can receive a signal from a whole decent size surface area, but there's only one path out. So once that signal is received, the neuron specifically sends it to where it needs to go. Okay? The neuron is the most specialized cell in the animal body. Okay? It is also the longest cell. Okay? A blue whale neuron can be up to 30 meters long. Okay? Giraffe about 5, and a human neuron between 1 and 2, depending on how tall you are. But also remember, even as long as the 30-meter neuron, the signal can still travel in less than a millisecond. It's amazing how neurons work. Alright, transmission of a signal down a neuron. It works a lot like dominoes. Okay? You start the signal. So you knock down the first domino. And what does that first domino do? It knocks down the second, and then the third, and then the fourth, and it keeps going down. Okay, so that triggers the signal. Then the signal moves as each successive domino is knocked down. Does one domino go here? Does the first one start here and then end up at the very, very end? No. The dominoes themselves do not move. But by the force of one falling and then bumping over the next, and bumping over the next, bumping down the next, the signal moves down like a wave through them. Okay? Then at the end, once all the dominoes have gone down, you still have to reset the system. You can't just do it again right away. You have to physically set back up all of the dominoes. This is kind of like how your neuron works. It does have to reset. Fortunately, it resets a lot faster than dominoes. Okay? All right. So there are protein channels in your neuron. Okay, remember protein channels are proteins that go transmembrane all the way across the membrane. They're going to allow materials in and out of the cell, usually materials that can't fit through the membrane on their own. Okay, the first one, like the one at the very tip of the dendrite over here, is going to open up, and because of changes that happen, it's going to cause the next one to open, then the next one, the next one, and the next one. So the signal moves down the entire neuron. This is an all or nothing response. Once the first one opens up, the rest of it's going to go. Okay? It travels like a wave, and it has to be reset before it can go again. Alright, so some things to remember about our cells. Your cells live in a sea of charged ions. Okay? Anions, remember, are negative, and there are usually more of those within a cell than without, because of the fact that your cells are full of proteins, and almost all proteins are negatively charged. Okay? And then there's also usually a lot of chlorine atoms inside of your cells as well. Cations, remember, are positive, and they're usually more concentrated in the extracellular fluid outside your cells. It's usually sodium that is the biggest one of those. So because of the fact that the inside of the cell is usually negative and the outside of the cell is usually positive, there's this difference in charge across the membrane. And that the membrane is said to be polarized, negative inside, positive outside, creates this charge gradient, and it literally stores energy like a battery would store energy. Okay, so your cells have an actual voltage to them that can be measured. And if you stick a little voltmeter into a cell, the difference in charge between the outside of the cell and the inside of the cell is 70 millivolts. Okay, so there actually is this buildup of charge. We call that buildup of charge the resting potential of a cell, because that's what it's like under normal circumstances. That's its normal potential. So the resting potential of a nerve cell is minus 70 millivolts. Okay. Now, how does a signal move down through a cell? Well, first of all, somehow the nerve has to be stimulated. And there are different things that are going to stimulate different types of nerves. Some of your nerves are touch sensors, so literally pressure. As you push your finger, you feel that. And the reason you feel that is because the neurons that are in contact with your skin are triggered by pressure. Okay? Some of them, like the ones in your nose, are triggered by a chemical. So when you smell something, you're actually getting small pieces of that thing, and the chemicals and the atoms that are in there trigger and open up or stimulate one of your neurons that then sends a signal to your brain. The neurons in your ears are also have a touch sensor, because remember that sound travels in a wave, and it's going to literally move your eardrum. So then you have nerves inside your ear that sense that movement, and that stimulates them. Different types of nerves have different types of stimuli. Once the nerve has been stimulated enough, so you may notice that somebody will come up behind you and kind of touch your back and you don't realize it, because it wasn't a hard enough pressure to trigger the neuron to actually send a signal to your brain. So the stimulus has to be large enough that it actually reaches what's called the threshold potential and makes the neuron actually go. Okay? And what that is, the actual going of the neuron, is when the very first sodium channel opens. Okay? So transmembrane protein in the neuron is going to open up, and sodium is going to come into the cell. Okay? Sodium ions are going to diffuse in because the inside of the cell is negative, and there's a big buildup of sodium ions outside the cell. So by pure diffusion, going from high concentration to low concentration, sodium is going to move into the cell. Okay? When all those positive ions come in, that is going to cause the charge of the inside of the cell to change. It's going to go from negative to positive. Okay? It's now going to be positive on the inside and negative outside, because all these positive ions went rushing inside. Okay? This means that the cell becomes depolarized. That's what we call it. It's like the first domino going down. Now what happens? How does this keep moving? Well, again, it's going to be by diffusion. Okay? Not only are the sodium ions here, they're going to all be concentrated because they just came in here, so they're going to start diffusing down the way. And as that positive moves down, as the positive charge hits the next voltage-gated channel, it's going to open up and more sodium is going to rush in. So as soon as this next membrane protein senses the positive charge inside the cell, it's going to open up. That's its stimulus that makes it open. It opens up and more sodiums come rushing in. Okay? And then, again, it's going to reverse the polarity and keep moving everything down. And it's just going to keep happening. Sodiums move down, open up a channel, more come in. They diffuse down, open up the channel, more come in. It just keeps happening all the way down the neuron. Okay, the second set. Once the sodium has started to move down the neuron, behind it, the neuron's going to start resetting itself. So before the neuron has even fully finished firing, the beginning of the neuron is already resetting to get ready for the next one. That's why you can feel a stimulus over and over and over and over and over again, because your nerves can reset very, very quickly, because before the first stimulus is even done being sent, the cell is getting ready for the next one. So how do they reset? Well, once the sodium ions start to move down, there's going to be another membrane channel. Okay, another membrane protein. There's a sodium one. Right next to it, there's a potassium one. The potassium one's going to open up. And now potassium is going to come rushing out of the cell, because there was a bunch of potassium in here, not much out there, so by pure diffusion, potassium is going to go rushing out of the cell. Okay? And this is going to reverse the charge back. So now the inside of the cell is going to be negative again, and the outside will be positive, because all of these positive ions left the cell. Okay? And that sets everything back up. Okay? So these two combined waves move down. First the sodium wave moves down, and then the potassium wave moves down. So fire the neuron, reset the neuron. It only goes in the one direction. Okay. So this impulse, this movement of the sodium ions down the neuron, we call this the action potential, because things are actually moving, action. Okay? And it can go from your fingertip to your brain in milliseconds. Okay? So the sodium channels, remember, are going to open up really, really quickly. So as soon as the neuron is stimulated, the sodium channels open up, sodium comes rushing in. The potassium channels open up much slower. Okay? So they open up slower because then that allows for the signal to go through, and then after the signal's past an area, then it starts resetting. That way you don't get reset happening before it's actually gone. That would be bad. But notice structure and function again. If it happened the other way around, if the potassium channels open first, you'd never get a message. So the structure of how this works meets with the function, the job it actually needs to do. So once a neuron has fired and then has reset its charge, it's positive on the outside again and negative on the inside, but the potassiums and the sodiums are in the wrong spot. So the nerve won't be able to refire the way that it should. So the sodium needs to move back out, and the potassium needs to move back in, so they're in the right spots for when the neuron tries to fire again. Okay? But both of them have to be moved against their concentration gradients. So this is going to require active transport, an actual pump. We call that sodium-potassium pump. Okay, sodium-potassium pump. Number one example used to explain active transport in every textbook, usually on every test. It's all there. Okay, it requires ATP. Remember, since active transport is going against concentration gradient, it requires energy to do this. ATP. So the way that this works is that 3 sodium ions are pumped out and 2 potassiums are pumped back in. Okay? And this is going to totally reset the charge and reset the concentrations where they need to be. Okay? It's going to get everything so it's positive on the outside, negative on the inside again, and ready to fire again. Okay, so it gets us back to our resting potential. Okay? So this is kind of this graph over here shows the voltage in a neuron is in one location as an action potential goes past it. So here's the resting potential. Remember, it's at minus 70 millivolts. Okay, some sort of stimulus reaches its threshold, which means that the neuron is going to start firing. That's going to cause depolarization. You're going to get your sodium and your potassium channels to open up, or sodium channels open, potassium channels close, and the charge is going to go from negative all the way up to positive. Okay. Then the sodium channels are going to close, the potassium channels are going to open back up, and the charge is going to go back down to negative. And it's actually going to undershoot, and it's going to get more negative than it was before. So as the cell is reset, it's going to go back up to the 70. So it goes down almost all the way to 80 as it's getting ready to be reset. So you can see this change in the charges in one location. All right, so that's how a neuron fires. But neurons fire really, really, really quickly. I mean, milliseconds to get from your toe all the way up to your brain, that's pretty impressive. And if it was going all the way down that whole neuron that reaches from, because there is one neuron that goes from the back of your heel all the way up to the top, to where your spinal column meets your brain, that's your sciatic nerve, it's a long distance. And if it had to go over literally every single millimeter of that neuron, it wouldn't be able to go very quickly. So our neurons actually have something called a myelin sheath on them. And this is going to help the signal go faster. Okay? So the axon itself, remember that's like the tail of the neuron, is coated with these things called Schwann cells. And Schwann cells make a myelin sheath. And they're going to insulate the axon kind of like the plastic on a cord does. Okay, remember the electricity is in the middle of here, and then this plastic helps insulate us so we don't get shocked by the cord. Okay, so what's going to happen is that the signal, the electric signal, the change of charge is going to jump from one spot to one spot to one spot. See, there's these spaces in between two pieces of insulation, and the signal is going to jump between those spots. And that's going to make it be able to move a lot faster. So see, there it is jumping. Okay, speeds it up from 100 all the way up to 150 meters per second rather than about 5 meters per second. I mean, 5 meters per second is still really, really fast, but 150 is way faster. Okay, so you can see here's the action potential happens right here, and then it happens again right there, it happens at the next node, and it goes from node to node to node to node to node. Okay? If a person has a disease called multiple sclerosis or MS, that means MS is an autoimmune disease. It means the body literally starts attacking itself. So the body's own immune system will attack its myelin sheath. And that means that this myelin sheath goes away. It makes neuron function much slower. It can make the edges of the nerve much more sensitive. So it can cause paralysis because neurons don't function the way that they should. It can also cause a lot of pain because sometimes they overfunction. It can be kind of tough. Okay? Now, what happens when a signal gets to the end of a neuron? It gets to the end of the axon, it goes to the synaptic terminals. Remember, this is the very end. Remember, I told you that one neuron could connect to another neuron, or it could connect to a muscle, or it could connect to the brain or the spinal cord, something like that. But here's the thing. Like a neuron and another neuron, or a neuron and a muscle cell, they're not going to actually physically touch. There's always going to be a space in between the two. Okay? So we call that space the synapse, or the synaptic cleft. Okay, it has to go from one to the next. Usually, in almost every location and every neuron, you've got neurotransmitters, which are going to be chemical signals that are going to be triggered to leave one neuron and go into the next one, or into the muscle cell, or whatever it is that's being triggered. Okay, so the chemical signal leaves, diffuses across that synaptic cleft, that actual opening. They are received by proteins on the next spot, and that causes a reaction then in the next spot. Okay, so action potential depolarizes this membrane down here, which opens up calcium channels, okay, which allows your neurotransmitters to actually be released. These are some neurotransmitters, and they move across the cleft. By diffusion, they're going from where they were highly concentrated to where there aren't any, so that's low concentration. They bind with these receptor proteins, and then they cause sodium channels in this now section to open up, and the next neuron can fire. Okay? It's kind of interesting. Your body takes an electric signal, literally the change in charge, and converts it to the chemical signal of a neurotransmitter. Okay? So here we go. So here's your once the signal is received at the next neuron, it's gonna, same thing, open up the sodium channels, and the whole thing's gonna happen again. Okay, so here's your neurotransmitter. This is acetylcholine. It binds with this receptor protein and causes sodium to come rushing into the cell, triggering this new neuron. Okay, and here we go again. Off we go. There are a lot of different types of neurotransmitters, and you don't really need to memorize these, but you want to just kind of look them over and be a little familiar right now. Acetylcholine is going to signal your skeletal muscles to actually contract and release so that you can move. Epinephrine is going to kick in that fight or flight response when you're nervous about something. Dopamine affects sleep, mood, attention, learning. Lack of dopamine, we've noticed, is associated with Parkinson's disease. We don't really know what is causing what. Like, do you run out of dopamine and therefore you get Parkinson's, or does something about Parkinson's stop your body's ability to make dopamine? We don't know what the cause is there, but we do notice that people with Parkinson's have a much lower amount of dopamine in their brain. Okay? And serotonin is another one that's going to really affect sleep, mood, attention, learning. Serotonin and dopamine are two of the ones that antidepressants can help regulate the levels of, especially serotonin. Okay? They are a weak point of the nervous system. So sometimes your body doesn't make the right ones, or doesn't make enough of them, but then also certain substances in the environment can mimic them And so they can block, like you can get something in that mimics a neurotransmitter, and then your body's responding to that rather than to the actual neurotransmitter. Some of them block neurotransmitters or break them down. So nitrous oxide, carbon monoxide, those are two examples. Carbon monoxide can shut down the neurotransmitters that allow you to do respiration, and then you run out of ATP and you die. Mood-altering drugs can also really impact your neurotransmitters. Caffeine impacts neurotransmitters. Okay. All these things. All right, so here's a little bit on acetylcholinesterase. Okay, it's an enzyme which breaks down acetylcholine. Okay, this enzyme, these enzyme inhibitors that stop acetylcholine from being broken down, it's a neurotoxin. Okay, it's going to stop your body's ability from getting rid of acetylcholine and stop your body's ability to stop your muscles from firing. Okay, and it's active in these different types: snake venom, sarin gas, insecticides, and they're going to cause the muscles to overfire. Okay, all right. So some things to think about as you consider nervous systems. Now remember, in the book there's a ton of stuff about all the different parts of the nervous system. Please do not focus on the parts of the brain. You do want to know the difference between the central nervous system and the peripheral nervous system. So central nervous system is your brain, brainstem, and spinal cord. Peripheral is all the neurons leading out. So you want to know that. But other than that, you're pretty much good. Okay? Okay guys, I want to show you a quick animation that's going to go over neuron function again in a little bit more detail.
A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. The cytoplasm just inside the membrane is negative in charge, and the fluid just outside the membrane is positive. The membrane is said to be polarized. Like a flashlight battery, the difference in electrical charge carries potential energy, which can be measured as a voltage. The oscilloscope shows this charge difference, called the resting potential, in millivolts. A neuron has a resting potential of about minus 70 millivolts. What causes the resting potential?
The plasma membrane of the neuron keeps large negatively charged molecules, such as proteins, inside the cell. Pumps and channels in the membrane regulate the flow of positively charged sodium and potassium ions. The neuron maintains a higher concentration of sodium ions outside the cell than inside, and a higher concentration of potassium ions inside the cell than outside. In a resting neuron, most sodium channels are closed, but many potassium channels are open. As a result, very little sodium can diffuse into the cell, but potassium ions can flow freely out of the cell, adding to the positive charge outside. The sodium-potassium pump actively transports more sodium ions out of the cell than potassium ions into the cell. This contributes to the buildup of positive charge outside the cell, while keeping the concentration of sodium low and the concentration of potassium high inside the cell. The diffusion of positive potassium ions out of the cell and the large negatively charged molecules trapped inside the cell cause the charge difference that constitutes the resting potential. We have looked at the resting potential in a neuron that is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus may change the permeability of the cell membrane to sodium or potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal called an action potential that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close in response to voltage changes across the membrane. When no action potentials are being transmitted, most of these channels are closed. An action potential begins when voltage-gated sodium channels open and sodium ions rush into the cell. The plasma membrane becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close, while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The plasma membrane returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in, and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. The efflux of potassium ions during the falling phase of the action potential restores the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting distribution of sodium and potassium ions.
Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
Alright guys, so that little video hopefully helps make the whole thing work a little bit more for you. You're going to spend some time doing an activity with this where you're going to be actually modeling the movement of ions in and out of a neuron. And hopefully all these together will make this whole process make sense. Have a good day.